Alright, hi everyone. Today we will be talking about brachytherapy, a medical physics technique. Brachytherapy itself is a subfield of radiation therapy, a form of medicine that harnesses ionizing radiation to treat cancers. The other branch of radiation therapy is external beam radiation therapy, or simply EBRT. As its name suggests, EBRT employs radiation sources exterior to the patient. Contrastingly, brachytherapy uses radioactive implants to deliver localized, conformal doses of radiation. This is the topic we will explore today. To understand brachytherapy, a brief review of nuclear physics is in order. Recall that any atom is composed of a dense nucleus of positively charged protons and electrically neutral neutrons, orbited by negatively charged electrons. At all times, the strong nuclear force works to hold the nucleus together, while Coulomb interactions between like-charged protons attempt to force the nucleus apart. When there is a disequilibrium of these nuclear forces, a nucleus will decay into a stable atom and emitted radiation. Radiation will impart energy to any material it passes through, iteratively decreasing its own energy. When radiation is so low energy that it can no longer pass through material, it is referred to as non-penetrating. When penetrating radiation has high enough energy to remove electrons from the atoms of the material it passes through, it is known as ionizing radiation. This is because the removal of electrons leads to the formation of ions. For instance, when gamma rays pass through skin, they ionize water molecules, leading to the formation of free radicals. This can result in DNA damage, loss of reproductive function, or even cell death. The study of the effects that ionizing radiation has on living organisms is known as radiobiology. It forms the theoretical basis for clinical radiation therapy. There are 9 different biological responses that individual cells can have to radiation. It may take multiple days and treatments before any of these effects is observable. Moreover, each cell can exhibit multiple of the 9 symptoms. There are also 5 population-level responses to radiation. They are referred to as the 5 R's of radiobiology and occur both during and after radiation. It is important to note that when we talk about a population in this context, we simply mean the irradiated volume of cells, which will inevitably contain both malignant and normal cells. Together, the 9 cell-level effects and the 5 R's can be condensed into 2 effective clinical outcomes. Either a cell dies as a result of radiation or it survives. Simply put, the goal of brachytherapy is to control which cells die. A successful treatment plan maximally kills tumor cells while sparing normal tissue. Brachytherapists attempt to accomplish this by fine-tuning 3 parameters: dose, fractionation, and source placement. Dose refers to the intensity of radiation that an area receives. It is measured in units of grays, where 1 gray is equal to 1 joule per kilogram. Doses are spaced out over multiple days. This is known as fractionation. For instance, a standard brachytherapy fractionation scheme is 2 gray per day delivered over a period of 30 days. Finally, source placement describes where in the body radioactive seeds will be placed. To solidify these concepts, let's now look at an application of brachytherapy to treat a synovial sarcoma. On the left, we see an arm with brachytherapeutic implants in place. The tumor has already been surgically resected or cut away, and brachytherapy is being used as an adjuvant therapy to irradiate any malignant cells not visible to the naked eye. This is crucial since residual tumor cells can result in tumor recurrence, metastasis, or radio and chemotherapeutic resistance. The white tubes running parallel to the arm are the catheters which radioactive seeds will be threaded through. The parameters we talked about on the last slide are shown here. This is an MDR brachytherapy with a fractionation scheme of 38.5 by 10 by daily. In other words, a dose of 3.85 gray will be given twice a day for 10 days. So far, we've talked about the very basics of brachytherapy, namely that clinicians fine-tune dosage, fractionation, and source placement to modulate the biological impacts of radiation. We will build on this understanding to discuss more advanced topics during the in-class presentation, such as applications of H&E and IHC staining to classify cancers into subtypes, and generation of isodose profiles via Monte Carlo simulation. Thanks everyone for watching, and if you have any questions, feel free to reach out via email.